My dear students, today's topic is how we observe the cell. When the cell size is very large, particularly in the case of egg, bird egg or ostrich egg, it can be seen by naked eye. In the largest cell is ostrich struthio egg. It can be seen by naked eye. When a normal cell whose size range from 3 to 5 micrometer or even the 10 micrometer, 1 to 10 micrometer. Particularly all eukaryotic plant and animal cell. All eukaryotic plant and animal cell. Eukaryotic cell. These eukaryotic cells generally we can see by compound light microscope. Compound light microscope. Here 2000 magnification, near about reach maximum 2000 times a object can be seen as magnification. Then the prokaryotic cell. The prokaryotic cell, that is the bacteria, their size starts from 0.3 micrometer, 0.3 to 5, 0.3 to 5 micrometer, comparatively smaller than eukaryotic cell. They are primitive. These are found, some amount are found in compound light microscope and some are seen by electron microscope. Where the magnification is about 2 lakh times. By this electron microscope we can observe mycovirus, mycoplasma. Mycoplasma is the smallest cell of animal kingdom or living world. The mycoplasma is the smallest cell. Its size 0.1 to 0.3 micrometer. Living cell. And virus is smaller than mycoplasma. Its size vary 0.02 to 0.2 micrometer. These virus, mycoplasma and bacteria all are seen by electron microscope. Then we can, in some cases, some larger cells, particularly found some algae or the single cellular fungi, the yeast, can be seen by here simple light microscope. Simple light microscope. Here about 200 times magnification may be occurred, and by this process we can see some large unicellular organism. From this observation, it is concluded that the cells size vary. Some cells are very large, some medium size, and some small, and some is the smallest. The smallest cell, that is the virus and bacteria and mycoplasma, can be seen by electron microscope. Some bacteria are also be seen by compound light microscope. Generally, they are all eukaryotic, and eukaryotic plant and animal cells are seen by compound light microscope. And some unicellular organism, even the egg of the human, can be seen by simple light microscope. The magnification, range of magnification are different in different microscope. Here, and what are the magnifying apparatus? Those are used in this microscope. Here only biconvex lens, single one biconvex lens. Here two types of biconvex lens are used. One type of lens used in the eyepiece, and second type of lens are used in the objectives. In two area, several lenses, convex lenses, are arranged in a specific manner, specific arrangement for the proper magnification. And third is the electron microscope. In the electron microscope, generally a electron gun is used. Here the light, here light is the source, here light is the source, but here source is the electron source. Electron gun is used. This electron ray fall on the specimen and reflect or transmit, and this direction of the electron flow is also regulated by some higher electric field. These electric field are created by some electric coils, where high amount of electric voltage differences are applied. These several coils are electrical coils are used. Those electrical coils create a huge amount of magnetic field. These magnetic field regulate the electron flow that fall on the strike on the specimen, and the specimen, magnified image of the specimen, we can see by this type of microscope. Then the question arises, then the question arises, number of cells in organisms. These organisms, some organisms contain a single cell. This is the unicellular organism. Unicellular organisms. Unicellular organism means contain only one cell. Only one cell. That is, those are found in amoeba. Example, amoeba, chlamydomonas. Amoeba, chlamydomonas. These are unicellular. Amoeba, it is irregular in shape. They have pseudopodia by which it can move from one place to another, and chlamydomonas. This is the cell of chlamydomonas. This is the chlamydomonas. This is the amoeba, and this is the chlamydomonas. Unicellular organism. Some organism, they can be considered the second type. Some organism contains 100 or 200 or 1000 or 10, but more than, that means more than one cells, but the cell number is not very high. Moderate number of cells. Organism having medium size, having medium number of cells. These organisms are volvox. Volvox is a algae that contain some chlamydomonas like cell organization. Group of cells. These group of cells are interconnected by means of some thread. Group of cell, and this is the can move. As a group of cell can move because they have locomotory organ, flagella. By this flagella, they can move from one place to another. In some algae, chlamydomonas, this is the volvox. Chlamydomonas, and this is the spirogyra. Chlamydomonas is the unicellular, spirogyra is a filamentous, but it is multicellular filamentous structure, and the cell contain spirally coiled chloroplast. These are spirogyra. These are also algae. Then the organism having huge number of cells. Multicellular, complex multicellular organisms. Third is the complex multicellular organisms. Like all higher plants and animals. Example, higher plants and animals. Particularly when we consider a human being as a more advanced organism, the body, when we consider our body contain human body, number of cells, human body, 37.2 trillion. Trillion number of cells present. Even the 100 billion nerve cells present in human body. And when we consider nerve cells, that is the neuron that communicate our body with our external environment, and different organs in our body are interconnected by neurons, nerve cells. Human body, nerve cells number 100 billion. Even when we consider 1 drop, 1 ml of blood that contain 5 to 6 million RBC, 1 ml blood contain 5 to 6 million RBC. That means red blood corpuscles. Red blood cells, this is the red oval, biconcave, these are biconcave cell, this is the red blood cell. 1 ml of blood contain 5 to 6 million. In one blood of cell, 6 to 7,000 WBC, 6 to 7,000 WBC white blood cells may be found. When you draw a WBC, these cells contain a multi-lobed nucleus. This is the lobe each nucleus have. Here 4 lobes are found. This is the neutrophil, a type of WBC. These are the general features the size of the cells. In living kingdom, animal kingdom as well as the plant kingdom, even the living world, when we consider the mycoplasma, virus and bacteria, different cells having different size. They are ranged from 0.02 micrometer to 10 meter. Very small size cell, 0.02 to 0.2 micrometer. These are the virus. What is virus? Virus is a crystalline substance having the character intermediate between plants, living organisms and dead organisms. They live in our environment without having any entity. But when these organisms enter in living organism, they replicate and reproduce. In this case, they show living activity. In normal environment, it have no living activity, but when enter into other living organisms, it replicate, reproduce, and showing and capable to reproduction and producing many offsprings. These are the virus. Very small size, and these can be seen by electron microscope. Suppose we consider a picture, a phage virus. Phage virus is a virus that infects bacteria. These are the one of the important virus is the phage virus, bacteriophage. T2 or lambda. Then after greater than the size of the virus, greater than the size range from 0.2 micrometer to 1 micrometer. These 1 micrometer range are found in the mycoplasma, gallisepticum. It is the PPLO. Greater than size than virus, but it is the smallest cell. Greater than the size of mycoplasma, the third virus. Then we consider the second organism, that is the mycoplasma. The range of the size of the mycoplasma 0.2 micrometer to 0.1 micrometer. Mycoplasma. Gallisepticum. This is also called PPLO. This size is smaller than virus. Their size 0.2 micrometer to 0.1 micrometer, and this is the smallest cell, smallest living cell, very small size cell. This can be seen by electron microscope. Then greater than mycoplasma, the third type, bacteria. Bacteria, their size starts from 1 to 2 micrometer. These are bacteria. Several bacteria, coccus, bacillus, different bacteria having different size, and these bacteria can also be seen higher resolution of compound microscope and as well as electron microscope. Suppose we draw a bacteria. This is the bacteria, and the center of the bacteria contain a nucleus-like structure. This is called nucleoid, and some flagella may be present from that emerge from the cell. They help them locomotion. Greater than the size, greater than bacteria that is found in eukaryotic plants and animal cells. Plant and animal cells. Their range from 10 to 20 micrometer. Most of the animal cells, higher animal cells and plant cells are eukaryotic, and their size is greater than bacteria. This is, for example, this is a typical animal cell, and this is a typical plant cell. Plant cell have a wall. Both type of this case have cytoplasm and nucleus that will be discussed later. Animal cell and plant cell. These are the size of the cells. Then we consider size is a very unique feature of cells. The smaller, if the cell having smaller size, it has greater efficient. It is greater efficient in case of its intercellular communication, as well as if the greater smaller cell having greater surface area, and they have the capability to absorb different nutrients. They excrete huge amounts of the toxic materials. Even they divide huge number of cells in a small time. This is the advantage of small cell. Suppose the size is very important when a large cell, suppose this is a cell having a diameter, breadth, length 2 millimeter. If the cube height, breadth and length both have 2 millimeter into 2. In this case, when we consider a cube, it has 6 faces, and each face, when we consider this face, its area is equal to 2 into 2, as the length of each arm is 2 millimeter. And when we consider this cube and it contains 6 faces, then it multiplied by 6. In this case, we can find that the volume, the surface area will be 2 into 2 into 6, 24 square millimeter. When we cut this cell and divide it into 8 equal half, when we cut this cell in 8 equal half, in this way, and cut this cell in 8 equal half. When we consider this size, as it is divided into 2, 8 half, that is why each length is transformed into the value 1. In this case, its value, when it is 78 piece, 8 equal half, and each arm having 1 millimeter in length. In this case, what happened? 1 length into 1 breadth into 6 face. How many number of cubes are produced? When a cube having 2 millimeter, its arm are divided into 8 pieces. In this case, into 8. In this case, the total surface area will be 6 into 8, 48. That is double of the surface area. That means when a large cell is divided into smaller parts, its surface area also increases. And when a cube divided into 8 equal parts, each part, when the total volume of total surface area of each equal cube we consider, then the total surface area will be increased into double. This is the increase the surface area. What is the advantage of this, of the increasing of the surface area? The advantage, first is the advantage. What is the advantage? Advantage of increasing surface area. First advantage is the cell to cell communication will be very functional. Cell to cell communication will be increased. Then second, the surface area for absorption and excretion is also be increased. The absorptive surface is also increased. As a result, when the surface area of a cell increases, different type of nutrients the cell can absorb easily. First point, it can absorb different type of nutrients, absorb nutrient. Similarly, excrete some excretory substance, excretion increased. As the cell contain their surface area increase, their internal cytoplasmic amount also be increased, and their metabolic activity and the physiological activity is also be increased. Physiological activity, is also be increased. This is the significance of the surface area.